tarde a todos. Vamos começar então o nosso primeiro colóquio da pós-graduação de 2021. Né? E é um grande prazer ter a professora Clarice Aiello hoje como a nossa convidada. Antes de passar a palavra para a professora Clarice, só lembrar os participantes o seguinte, que essa apresentação está sendo gravada e ela vai ser disponibilizada publicamente. Então, por favor, né, mantenham os seus vídeos e áudios desligados. E caso desejem se identificar, vocês automaticamente concordam com a divulgação pública da sua manifestação. Essa mensagem está no chat, caso alguém queira ler de novo verificar. A gente começa a apresentação e, em geral, a gente faz as perguntas, deixa a parte de perguntas para o final. E, em geral, a gente tenta usar aquele sistema de levantar a mão ou, quem não quiser se pronunciar, simplesmente coloca a pergunta no chat que a gente lê. Tá bom? Então, Clarice, muito obrigado pelo convite, muito por ter aceito o convite e por estar aqui hoje abrindo o nosso colóquio do semestre.
Obrigada pelo convite. Estou muito feliz de estar falando com todos vocês. Eu nasci e cresci em São Paulo, minha família é de Jundiaí, pertinho aí da Unicamp. Então, é super legal estar falando com vocês aqui e espero que a gente tenha uma boa discussão no final. Vão anotando aí as suas questões, tá? Vocês vão me desculpar que eu não sei fazer a apresentação em português, então, para ficar um pouquinho melhor, eu vou fazer em inglês, mas é inglês com sotaque brasileiro, então eu acho que vai dar certo. Mas no final, eu acho que dá para a gente fazer as questões em português ou em inglês, então, vamos lá. Tá. Vamos ver se está funcionando. Funciona, ok. I like to call myself a quantum engineer. This means that I build experiments to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics as opposed to the laws of classical mechanics that rule everything big around us. So the title of my colloquium is a little bit weird, right? It has spin physics in it, so yes, I think as a quantum engineer, I know a little bit what spin physics is, but there's also the name biosensing in the name of my colloquium. And I have to admit that I never took a biology course after high school. So what do I want with this? So before I explain where I'm going, I really need to tell you where I come from. So we're going to start talking about hardcore quantum mechanics, and by the end of the talk, we will have talked about things of biological relevance, such as organismal migration, how our cells respond to stress and to radiation, and I hope to have motivated a little bit how I think we can learn with nature to build better technologies. And that's because I'm a quantum engineer who's interested in how quantum physics informs biology at the nanoscale. So I'm going to open a parenthesis. This parenthesis is going to be closed, but there's a reason why it's there. And I'm going to start everything by saying that I think humankind is obsessed with measuring things better because this means that we understand nature better. We can think about measuring better frequencies with, for example, this atomic clock. This atomic clock lets us define very precisely what a second is, based on atomic transitions. We can think about measuring better magnetic fields so that the image of your baby is better resolved. And yes, this is a magnetic resonance image of a baby inside the mother's belly. And we can also think about measuring better accelerations, for example, so that your gaming experience is enhanced. Those tiny little accelerometers are inside our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity that you want to measure is very, very small? I'm going to argue that in this case, you need a tiny little sensor that can measure tiny little things. If the sensor is tiny, let's make it very tiny. Let's make quantum. There's actually a whole body of mathematical arguments that prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the outcome of your measurement. I would like to tell you a little bit about a technological quantum sensor that I worked with in the past. And this technological quantum sensor has the form of a single spin in diamond that can be put to use as a very sensitive magnetic field sensor. So this is what I'm promising you, and I'm sure most of you are already familiar with the concept of spins, but maybe there are some freshmen there. So spin is a merely quantum property. It doesn't have a real classical equivalent that measures how some elementary particles, such as electrons, how well they interact with magnetic fields. Physicists usually depict spins, and you should be looking at me now, with an arrow, right, that can be up and down, just signify two different energy levels of this merely quantum property called spin. So the spin that I played with arose from a tiny crystalline defect in diamond. Okay, so this crystalline defect in particular is one out of many color centers. The color centers in diamonds are the lattice defect, the lattice imperfections in diamond that actually give rise to the color of those nice diamonds that you see in at the bottom of the slide. The particular defect that I worked with is called a nitrogen vacancy center. It arises naturally, but it can also be engineered when a vacancy, a missing carbon atom, sits nearby to a nitrogen, which is one of the most commonly occurring substitutional impurities in the diamond lattice. When those two things get together, it's a mess of electrons, right? There's like the unpaired electrons close to the vacancy. There's like the extra electron from the nitrogen. There might be extra charges around. So it's really like an atomically sized mess. But something very nice happens when you start calculating the quantum mechanical energy levels of this mess. And it turns out that the quantum mechanical energy levels that you need to represent this defect are very similar to the quantum mechanical energy levels of one single electron spin. So it's very convenient because we can look at this and we can think about one single electron spin. And the way that we find those defects in diamond, I mean, everything is again, it can be done at room temperature. And the very first diamond that we worked with was some discard from some person doing jewelry. So we take a slab of diamond and we put it on top of a moving stage, okay, as we shine laser, okay, and we start moving the sample back and forth. The idea is that once the sample gets to a point where we find one of those defects, it's a color center, so it will absorb light and then fluoresce. And this light can be collected in the lab. In the lab, we use a laser, but just because it's more efficient and more convenient, but it doesn't need to be a laser. And that's also why we can see the colors of the diamonds like with regular light. But this is how it looks in increasing zooms. Okay, so at the bottom right here of the picture, you see one blob. Okay, that actually it's super interesting. The defect itself is atomically sized, but this blob is diffraction limited. But it's still very impressive because if you look at this, you're actually looking at the fluorescence of an effective single spin. But it gets even better. And that's because, for reasons that I won't have time to explain, just by looking at how strongly this blob is fluorescing in your computer screen, you can actually know if the spin is up or down. This defect has in jargon, quantum state dependent fluorescence intensity. And this will come much later in my talk as well. But this is what we use to read the state of the spin in our experiments, intensity of fluorescence. But I promised that we could put it to use as a very sensitive magnetometer, and here's how we do this. 0 and 1 there just represent the two different energy levels of the spin. Their energy difference, their resonance, is in the microwave, about 3 gigahertz. And it turns out that the state that I call 0 there is insensitive to the presence of magnetic fields, whereas the state that I call 1 is sensitive to the presence of a magnetic field, so that if there's a magnetic field around, state 0 won't care, and state 1 will be sort of promoted to a new energy level. And you might remember, or you might have not seen it yet, but this tiny little detuning delta, this shift from resonance, is proportional to the intensity of the magnetic field via something called the Zeeman interaction. So, and that's super important, and this point doesn't get made many times. The problem of measuring a magnetic field is then mapped onto the problem of knowing how to measure a detuning delta from a non-resonance. That is the difference in energy between 0 and 1 in the absence of magnetic fields. And measuring detuning's delta from resonance is something that engineers and physicists have known how to do for many, many decades. So this tiny little spin can measure magnetic fields because the experimenter can measure detunings caused by the presence of magnetic fields onto this two-level quantum system. But there's a catch, and some of you might be familiar with it. Everything that starts quantum dies classical. This is why we live in a classical world. So this modality of using the NV center as a very good magnetic field sensor is only valid while its quantum properties last. While this spin system is well described by the laws of quantum mechanics. For those of you who have taken quantum mechanics, this process can only be used while there is still phase coherence for a superposition between 0 and 1. So these so-called coherence times for this technological quantum sensor at room temperature and in the solid state matrix for a crappy diamond is about 2 microseconds, and it can actually be engineered to be much, much longer. And this in the field that is known as quantum sensing and quantum computing at room temperature is incredibly long. 2 microseconds is super, super long. So what we want to do then is actually experimentally try to keep this electron spin in a good quantum state so that we can play with it as a good quantum sensor for longer, right? In the same way that if you have a swing, right, you want to kick the swing at certain intervals so that it can keep going. And the idea here is that if we can acquire a signal with this coming from this system for longer, if we Fourier transform the signal in time, we get a better resolution in frequency, right? And the way that physicists kick those systems so that they maintain their quantumness is with engineered electromagnetic fields. Okay, and this is of course started probably with nuclear magnetic resonance and Rabi experiments, Ramsey experiments from the very beginnings of atomic and nuclear physics. So before I give you one example of what it looks like in the lab, I just want to give you the big picture, okay? The big picture is this. The big picture is having a tiny little sensor that you can bring in close proximity to a tiny sample, and then you can measure, say, magnetic fields caused by this tiny little sample or by spins in this tiny little sample, because you can measure the tunings that the NV center is going to sense. So this is the big picture. Okay, now I'm going to go to like my most technical slides just to give you a flavor of what we do in the lab in this field of quantum sensing. So The particular type of electromagnetic pulses that I used to keep my spin kicked was a sequence of pulses known from nuclear magnetic resonance that is called Rotary Echo. And it's depicted to the right here in nuclear magnetic resonance language. The interpretation of this is as following. You are applying a continuous wave microwave, like on resonance between the 0 and the 1 states. It's continuous, and at particular intervals that the experimenter can choose, you are actually flip the phase of the pulse that you're exciting your spin with. So you're always exciting with microwave, and you phase flip this microwave at certain intervals. Okay? And this sequence is known from nuclear magnetic resonance, and it's known to correct for driving field imperfections, for microwave imperfections. And it's also known not to correct for detunings delta from resonance. And if you remember my last slide, this is exactly what you want to be able to measure. You want to measure detunings. And I won't have time to prove here, but by choosing the time that it takes between phase flips, you can actually give protection to the spin for different kinds of noise. Okay, but this is sort of what it looks like experimentally. We have a signal as a function of how long we apply the sequence of pulses for. And this signal is just like a normalized intensity, right? Say, if you are at the top here of the signal, you are, say, in the zero state. If you are at the lowest point here, you are in state 1, and everything in between means that you are in a superposition state between 0 and 1. We spent a lot of time applying something called average Hamiltonian theory, and now we understand to first order what the signal, how is the signal originating from the pulses, and the signal has a particular dependence on the detuning delta that we want to plot. So if we Fourier transform the signal, we get something that looks like a spectrum, and it's not super easy to interpret, but I tell you that with this spectrum there, we are measuring magnetic fields of about 1/10 of the Earth's, okay, which is very, very tiny. Again, not important. The importance is that we have this big picture here of a technological quantum sensor that works at room temperature and in noisy environments. So don't forget that I have just shown you a quantum sensor that works at room temperature and in noisy environments. Now I'm going to close the parenthesis and talk about what I really want to talk to you about. So until a couple of years ago, the sensor that I just described and these sensors there that you see were the type of sensors that I was used to dealing with. But at some point, after talking to a lot of people, I realized that, well, sometimes nature, nature has had millions of years to optimize their own sensors. And many times, natural sensors outperform humankind-made sensors in the craziest ways. Not all of those sensors might be quantum. In the past, I worked with different kinds of sensors. I worked with a light sensor, which is not quantum, but I also worked with a magnetic sensor, which I'm going to argue might be quantum. Okay. So in order for us to start talking about the biology, we need to talk about the biology at the nanoscale, namely the chemistry. So it's known in a field, it has been known for many decades in basic chemistry. It's a field called radical chemistry, that magnetic fields can alter the final products of a class of chemical reactions that involves unpaired electrons, involves unpaired electron spins. Okay, and in the slide there, I say involving spin correlated electrons. So for those of you who have taken quantum mechanics, by spin correlated electrons, I mean that they're entangled. But again, that's not super important. So, and now you should be looking at me because I bring my props. I travel with my spins. I never part with my spins. I'm going to show to you how this happens in basic chemistry. Okay, here's what's known to happen in many chemical reactions in the gas phase, in the solid phase, in the liquid phase. Imagine that there are two electron spins at, say, the outer shell of a fluorescent molecule. Again, fluorescent molecule is a molecule that upon photon absorption, there's enough energy for one spin to be promoted, and then it de-excites, emitting energy, fluorescence, right? So in the presence of a photon, this guy will go up, emit a photon, down, and go back. Except that for some processes, upon photon excitation, instead of going down via fluorescence, sometimes this guy hangs around here, and sometimes it meets another unpaired electron that is in the reaction. And I'm told by chemists and biologists that free electrons are actually not that hard to come by. So in the absence of loss, photon absorption is spin conserving, right? So that when this guy meets with this guy, they need to be in a singlet state because spin is conserved, and those guys were in a singlet state. And I didn't mention, but they are in a singlet state because of the Pauli exclusion principle that says you cannot have two electrons like this, right? So they're in a singlet state. And so that in the absence of any other loss, when these guys meet, they start in a singlet state. And we're not told this very often, but a singlet state is a non-trivial quantum state, is an entangled state. Okay, but this is going to ensure that at the beginning, they have a particular relationship so that they will interact in sort of a strong way. So here's that. The second fact that I want you to know before we talk about mechanism and external magnetic fields is the fact that some chemical reactions depend on spin. This means that, and in fact, they depend on those two spins. Some chemical reactions, they continue through one macroscopic, through one pathway, if the spins, and I'm going to use a jargon, are measured by the environment to be in a triplet state, pointing in the same direction. And some chemical reaction, and the same chemical reaction continues through a different branch if the spins are measured by the environment to be in a singlet state. Okay, so a fast process of interconversion between singlet and triplet states at the top of a chemical reaction might alter macroscopic final products of the chemical reaction. Okay, and those final products might be produced much, much later than this fast interconversion. If you have heard this nomenclature, coherent process that occurs at the very top of the chemical reaction. Okay, so this is how a spin change here might give rise to very big changes in chemical yield, or like it might make a difference macroscopically. The last thing I want to say is that, which is the last piece of the puzzle that I'm trying to explain, is that those two spins, they usually, and for the external magnetic fields that we care about, they experience different local magnetic field environments. So this guy here is more or less strongly hyperfine coupled to nuclear spins in the molecule where it came from. So this guy here mostly sees the magnetic field caused by the nuclear spins where this guy came from. And this guy here is sort of free. Okay, so it doesn't have any other couplings. So in the presence of a weak magnetic field, such as the magnetic field of the Earth, this guy here will see this magnetic field of the Earth, and this guy here will see the magnetic field of the Earth, plus the hyperfine field, which is way stronger. So this guy here almost doesn't care. for the external magnetic field, but this guy does. Okay. You also know that spins, they precess around magnetic fields depending on the strength of this magnetic field, right? So this guy is going to be, for weak magnetic fields, is going to keep on precessing, but different external magnetic fields will actually make this guy here is doing all the job, precess at slightly different frequencies. That depends on these external magnetic fields. This means that depending on the magnetic field, this spin is going to pick up a different phase with respect to this spin, so this guy here, as they precess, and this phase depends on the external magnetic field. Effectively, what this means is that different magnetic fields felt mainly by this guy is actually going to influence the probability of us finding the spins in a triplet or in a singlet state, because the phase that those two pick relative to each other is going to translate into a probability of measuring the spins in a singlet or in a triplet or in a singlet state. Okay, so this is how it is explained in basic chemistry that magnetic fields can alter the products of this class of chemical reactions that depend on light and that involves unpaired spins. And this has been demonstrated at room temperature, in solution, in the gas phase, in the solid state, down to magnetic field strengths of the magnetic field of the Earth, right? And the magnetic field of the Earth is weaker than the magnetic field that you sense when you hold your cell phone close to your face, right? Which is crazy small. So the relevance of this to what I want to tell you today has to do with birds. We're going to start talking about birds, but I don't. At some point, I'll stop talking about birds, but I'll have to start talking about birds. It's known since I think the 60s that birds, turtles, and butterflies, and other actually complex organisms, that at least is a partial cue that they use the magnetic field of the Earth to migrate, which I think we all agree is tiny, right? So how do they do this? In the 80s, a theoretical biophysicist proposed the following. Imagine that this same type of spin-dependent chemical reaction were to be happening inside organisms at under physiological conditions. Okay, if this is the case, then organisms might be able to sense magnetic fields to the extent that they can sense different physiological concentrations of products coming from these spin-dependent chemical reactions. Okay? So this was in the 80s. Since all those chemical reactions, most of those chemical reactions that are spin-dependent depend on light, people started looking for things in the retina, say, of migrating birds that could sustain a spin-dependent chemical reaction. So they started looking for photoreceptors, for proteins, say, in the eye that absorb light and then fluoresce, right? Just because the reaction kickstarts with the absorption of a photon. And at that point, the only animal photoreceptor that was known to sustain spin-dependent chemical reactions was this photoreceptor, this protein. It's a flavoprotein. It has a flavin called cryptochrome. And actually, cryptochrome is super cool because it's not only present in our eyes, in the eyes of migrating birds, in the antennas of migrating butterflies. It's present in all our cells because it's responsible for circadian regulation of our bodies. Okay, but that's how all of this field sort of started. And throughout, I don't know, 20, 30 years, there have been many experiments that link, even if partially, magnetosensing to cryptochrome. All those species there, they produce cryptochrome, and the organisms boxed in red are organisms for which there is some evidence that magnetosensing might be related to cryptochrome. And yes, humans are right there. So my point here is that I really believe that evidence for cryptochrome-based magnetosensing is really, really widespread, but unfortunately at very disconnected length scales. Okay. On the tiny side of the length scales, as you can see in the left of the slides, in the chemical side, right, you can do, for example, experiments with cryptochrome in vials. Okay, you do experiments with vials. One of my favorite experiments, for instance, is researchers recorded the fluorescence of the cryptochrome as a function of the time during which we shone laser of an appropriate frequency to be able to give this first kick onto the cryptochrome, right? So fluorescence as a function of laser. So the first thing that you see is that this fluorescence goes down. We're killing the chromophore, okay? We're breaking the thing, like frying the thing inside your vial. But that's okay. I mean, it's not okay, but it's acceptable. But the cool thing is that as the experimentalists pulse magnetic fields on and off, and this has been repeated many times to show that it's not an artifact, what you see is that the fluorescence sort of gets modulated up and down following the magnetic field. And that's those tiny little waves up and down here that you see. Now, I would like to bring your attention back to what I was talking about, the spin sensor in diamond, right? Just by looking at how that spin defect, how that blob was, how much that blob was emitting light, we could know if the spin was up and down, or up or down. Here, it's actually the same thing, and that's very convenient experimentally. Just by looking at how strongly cryptochrome is fluorescing, you can have an idea, well, you can actually quantify if the chemical reaction continued through the singlet branch or the triplet branch, or which proportion, like say, for a certain magnetic field, 30% of the times it goes through one path and 70% of the time it goes through the other path. So in direct correspondence to what I said about the diamond spin. The other cool thing is that there's partial evidence that these spins in cryptochrome at room temperature in solution can be quantum, can have coherence times up to about 1 microseconds, again, which is on par with the 2 microseconds that I quoted for the spinning diamond. So there's no question that chemically, this protein, and actually many others, I hope we'll talk about those at some point, but many other proteins, they are behaving as tiny quantum sensors. Okay. For the magnetosensing problem for navigation, the next length scale for evidence, unfortunately, is for things with like whole birds, whole flies, okay, what you see to the right of my slides. For instance, flies can be trained to find food in the presence of a magnetic field. Then the researchers knock out the cryptochrome gene out of the flies, and the flies can no longer find food via the magnetic field. In another experiment, the researchers put back human cryptochrome genes in the flies, and the flies are back to finding the food via the magnetic field. There are many things that are actually consistent with the spin hypothesis, but it's very hard to say that, well, the bird is doing this because the bird is migrating because there's a quantum process going on, right? It's people, the chemistry people are not convinced, the animal people, the whole animal people are not convinced. So there's a mismatch in what people believe or not believe. So here's what we want to do, we're starting to do now in our group. We want to bridge those length scales. What we want, what we are building our lab, are like it's like a glorified microscope with coils. What we want to do is like the same type of chemically inspired experiment, or another word for it, quantum sensing like experiments that people do in proteins, but say inside single cells or inside a couple of cells, to see if we can actually go one step further, just the chemistry, and try to see if we can have evidence for what I'm calling living quantum sensing in vivo, right? So again, I'm arguing that spins in proteins, some spins in some proteins are really acting as quantum sensors that work at room temperature and in noisy environments. So that what we're doing is we're taking technology from quantum sensing and atomic physics and start applying them onto those spins in biology, treating them as bona fide quantum sensors. So, but again, this is, I need to stop talking about birds, because the most exciting thing for me is that, again, there is evidence that this very same physics of magnetosensing, of spin-dependent chemical reactions, might be behind many biological phenomena, such as regulation of reactive oxygen species, which are the things that don't really make our cells happy, right? That's why we take antioxidants. They might be affecting metabolic processes, such as respiration rates. They can alter spins, are consistent with result, as explained. Spin physics is consistent with explaining results such as differences in cell growth, differences in say how much DNA gets repaired, and even things related to embryology. So I have a friend in Munich, and what he does is, like, he's a precision measurement physicist, and for a living he builds hypomagnetic chambers, and then he puts like cold atoms experiments inside, and inside his hypomagnetic chambers we have a residual field of about like a nanotesla, okay, which is tiny. Again, magnetic field of the Earth is 50 microtesla. Residual field inside his super hypomagnetic chambers, 1 nanotesla. Just for kicks, what he and another group in Mainz, both groups of physicists, so they're not biologists, but what they did was to, for 2 days, grow tadpoles inside these chambers under 2 conditions. One with just the residual nanotesla level magnetic field, and ones with an internal applied magnetic field that was the same as the magnetic field of the Earth. While the experiment with this artificial magnetic field of the Earth, the tadpoles were okay. For the experiments where the tadpoles grew under hypomagnetic field conditions, 30% of the tadpoles were sort of malformed. They macroscopically, they looked like weird. And again, this is macroscopic. There is no direct evidence that this has to do with the spin phenomena, but given the strength of the fields needed for malformations to arise, my best educated guess, and that's one of the things we want to do in our lab, is that this is indeed mediated by spin. So so this field is sort of called like quantum biology, and I really think that the bottleneck of quantum biology nowadays is like high-end, high-tech, quantum-inspired instrumentation, right? So when I tell people that I'm going to build a microscope, our microscope is not going to look like the picture on the left. It's going to be like an optical table with plenty of mirrors, electronics, right? So our idea is really to go as high tech as we can be, to either confirm or refute whether quantum physics might play a role in biology. We're super delayed. We started here at UCLA 1.5 months before the shutdown. So we're super delayed, but we're building things right now. Our lasers are on the table, but we're going slower than foreseen. but the other thing that I wanted to share with you about this project is that figure 1 in our papers are like what we expect to get from our experiment. So the experimental approach, and this is sort of sometimes different from what people do in biology. Our experimental approach is going to be driven by mathematical models. So let me give you one experiment. Now let me come back. So what we do here is we can make simple models that even us as like humble experimentalists can deal with. So we take information, say, about the photophysics of the protein that we're interested in, and we can actually make a open quantum systems, Lindblad sort of evolution of our quantum systems, which in this case is modeled by two, and this is like the crappiest way to model what's happening. There's many theorists doing way more sophisticated models, but what we do to have an idea of what we expect from our experiments is like we model two electron spins that start entangled, but otherwise that do not interact. And on top of that, one of those electron spins has a hyperfine interaction with one nuclear spin. So it's a 3 spin simulation, and that you can start to make some predictions. So I won't have time to explain everything, but I would like you to look at this top right picture here, okay? And this is one of the predictions that that we're dying to confirm or refute experimentally. So there, in this top right plot, what I plot is the magnetosensitivity of this protein called cryptochrome as a function of the strength of an external magnetic field. And magnetosensitivity, I mean that given a magnetic field strength, how well can the protein distinguish between like chemical pathways? For instance, if for a certain magnetic field, 50% of the times the protein, the chemical reaction continues through the triplet branch, and 50% of the time it continues through the singlet branch, this means that it's sort of random, right? Which way it's going to go. On the other hand, if for another magnetic field, 95% of the time the chemical reaction continues through this branch, and 5% of the time it continues through this branch, well, it's like there's a chance that there's a chance that that things can be distinguished because there's a lot of things that go through one pathway. So this is magnetosensitivity as a function of the magnetic field strength. The first thing that you see is that this curve, in theory, in our simulations, and we're not the only ones to have done those simulations, the first thing that you see is that this curve is not monotonic. It goes up and down. By, say, 10 or 20 times the magnetic field of the Earth, which is still small, the magnetosensitivity has gone down, which is understandable given our model, because as the magnetic field gets larger and larger and overpowers hyperfine interaction of this guy here, the local magnetic field of those two spins becomes sort of the same. So changing the magnetic field does not really help the spins picking up a phase as they precess. So this we understand. The other thing that I find cool is that the peak of the magnetosensitivity, the exact peak has to do with the model parameters, but for good parameters, the peak of the magnetosensitivity is very close to the magnetic field of the Earth, which again might be a coincidence, or it might be that these proteins were evolved to be at its most magnetosensitive under the environment where it evolved. right? And we think that working with chemists who can do directed evolution of proteins, we can check if this is a coincidence or if it's an evolutionary trait. So again, as I mentioned, and I'm almost finished, we really want to check whether quantum mechanics is, well, being used by nature, okay? But even if it's not, right? Even if it's not being actively used, we know that those proteins have, and other things around, like at the nanoscale in biology, there are many things that have like quantum knobs, right? So even if those things are not used by nature, I mean, we want to either establish or refute, right? But even if those things are not used by nature, the knobs are there. Can we start tapping onto those quantum knobs to say to therapeutic advantage? Can we start, say, messing up with spins in reactions to regulate reactive oxygen species, regulate inflammation, to sort of drive physiology towards treatment of diseases, for example, right? Can we bias a chemical reaction to go one way or the other way? And also, like, can we learn with nature, right? If nature is using those quantum knobs, it's probably doing so in a very optimized way, right? So can we learn with the strategies, with the optimization strategy, and nature probably knows very well how to deal with noise, right? So can we learn with nature how to optimize quantum sensors, quantum computers? I'm not saying let's replace Google's beautiful superconducting quantum computer with molecules, but maybe there's some strategy that we haven't thought of yet that can be applied on top, like as a control to Google's quantum computer, that could make them more resilient to noise. And the reason why we don't have quantum computers working today still is because everything that starts quantum dies classical. So there's a lot to learn on how to minimize the quantum nest quenching noise of interactions with the environment. And actually, we're not the only people who are trying to use like very high-tech experiments to deal and understand biology. There are many other people that are starting to do this too. It's if you look at the dates here, it's 17, 19. It's a field that is just starting, right? And I'm not endorsing nature, but I'm endorsing discovery of nature. I really think that interdisciplinarity is going to take us very far, right? In the near future. And I hope you agree that we made the cycle from quantum mechanics to biology and maybe to quantum mechanics again. I would just like to finish by thanking these brave people that are the reason why I wake up every morning, and they were super brave to start a lab during a pandemic in a subject that remains controversial, right? With I'm super proud. They are building this crazy infrastructure, and yes, we're always looking for collaborations for interested students. And if you're interested in quantum biology, the good news is that the National Academy of Sciences in the US, okay, has just sponsored the beginning of this month a workshop on that had quantum biology in it. So if you register, I think you can still see the recordings, right? So it's starting to mature as a subject. And oh, another thing, and if you're interested, since April of last year, I've been running weekly quantum biology meetings on Tuesdays at 7 a.m. in my time, which is 11, which would be 11 a.m. Your times. Contact me if you would like to be included to see what those are. And that's all that I had today, and may the quantum be with you. What are your questions? Silence.
Hi, muito obrigado, Clarice. Foi um seminário bem interessante, bem inspirador. Eu achei bem interessante essa motivação. Achei muito interessante. Então, a gente pode fazer perguntas de duas formas, né? Ou fazer se vocês querem falar diretamente com ela, fazer a pergunta, né? Ou pelo chat aqui, o que vocês acharem mais confortável para vocês, né? Então, está agora aberto, se vocês quiserem fazer perguntas.
Manda.
Orlando, já tem uma pergunta no chat.
Aqui tem uma pergunta, não sei se você consegue ler, senão eu vou ler para ti.
A biologia quântica já possui explicações do porquê o processo de fotossíntese possui uma eficiência energética tão alta. Então, quando as pessoas falam de biologia quântica, elas falam de duas coisas, né? Falam de fotossíntese e falam de migração de ave, né? Primeiro, assim, eu acho que tem que democratizar um pouco, que assim, eu estou convencida que fenômeno quântico vai muito além de fotossíntese e de aves. Se der tempo, eu explico. Eu posso falar um pouquinho mais. Mas sobre a fotossíntese, né? O negócio da fotossíntese é o seguinte, que só para, não sei se todo mundo já ouviu falar disso, quando a planta, né, absorve um fóton, né, cria uma excitação, e o negócio é que, com uma eficiência muito grande, esse fóton é transmitido para o lugar onde ele tem que chegar, né, para começar o processo. E o que se pensava é que o jeito de fazer esse processo tão eficiente fosse o fato de que o fóton tem que vir daqui, a excitação tem que vir daqui até aqui, né? E pensa num chão de ladrilho, só tem, sei lá, uns 7 ladrilhos para o negócio pular de um lado para o outro até chegar aqui, um número limitado de ladrilho. No começo tinha se falado, se pensa ainda mais ou menos, que essa excitação pode explorar todos os caminhos possíveis pelo ladrilho ao mesmo tempo, né, um tipo de uma superposição, para chegar ao outro lado. Né? Isso é o negócio original, né, que se falava que teriam esse processo de coerência. Eu não sei aonde fotossíntese não é o meu negócio. Tem gente até hoje falando que é uma coerência só clássica, tem gente que ainda fala de coerência que é quântica, né? E eu não sei, é um negócio de debate, só que tem gente, uma outra parte desse negócio da fotossíntese que a turma não fala muito, mas que eu acho que tem com certeza, né? Que é o negócio de noise-induced transport, que é assim, que às vezes o fato, um acoplamento da excitação com o environment, né, com as coisas ao redor, ajudam o transporte, né, que mesmo que o ruído, a existência do ruído pode ajudar a transportar a excitação. E foi feita uma experiência com íon, né, que é muito legal, que é meio que para demonstrar isso, que eles usaram esse experimento com íon para motivar que o mesmo podia estar fazendo na fotossíntese, né? Então, tinha 2 íons, né? Um que estava com o spin para baixo, o outro estava com o spin para cima, e eles queriam transmitir a informação desse íon para esse íon, ou seja, eles queriam fazer o flip assim, né? E o que os pesquisadores descobriram experimentalmente é que, assim, se você fazer o tuning do environment, né, para que ficasse perto de umas frequências de vibração dos íons, que esse environment podia ajudar o flip ocorrer, né, que é uma demonstração desse noise-induced transport. que é outra coisa que se fala. Isso eu boto fé no negócio da fotossíntese.
Tá. Tem uma pessoa aqui no chat que fala, Pedro Alvarez, pode abrir o microfone e fazer a pergunta, por favor? Pedro?
Vai lá, gente. Vai lá, gente.
Oi, estão conseguindo me ouvir?
Oi, Pedro, sim.
Oi, Clarice, eu queria dar parabéns pela sua palestra, que ela foi muito boa. Eu queria só também falar que eu comecei meu mestrado agora e a gente está trabalhando nisso também, teoricamente, teórico, em magnetorecepção aviária com baixa sensibilidade de luz, e tentando usar o que a gente descobrir para fazer aplicações tecnológicas também, mas tudo basicamente na teoria.
Vamos falar? Manda um e-mail para a gente marcar uma conversa. Mas fale, continue.
Sem problema, eu queria mesmo te mandar um e-mail, porque eu acho seu trabalho muito legal, eu gostei muito, fiquei muito animado. E eu queria te fazer algumas perguntas sobre como vocês tratam o criptocromo no laboratório. Eu queria, na verdade, perguntar se a reação depende do molecular pocket, né? Porque a reação ela só depende das duas moléculas, a flavina e o que você está usando de compar. E eu queria saber se daria fazer só com as duas moléculas mesmo, ou se ela depende do molecular pocket do criptocromo, e também se você tem que manipular de forma que todos os criptocromos que você está analisando estão na mesma direção, ou se eles podem ficar aleatoriamente.
Então, várias partes da sua pergunta, né? O Pedro, né?
Isso.
O que o Pedro está falando é que, assim, vê se é isso mesmo, que aparentemente a razão pela qual os spins no criptocromo são coerentes por um tempo relativamente longo, né, é porque tem um lugar que faz o shielding dos spins do environment, né, que protege os spins de outros spins dos elétrons de outros spins, né, e das coisas ao redor. Então, eu acho que o Pedro está perguntando é assim, se a gente tirar esse pocket, funciona? Eu acho que foi feito, eu tenho certeza que foi feito experimento sem o criptocromo, só com a flavina e o outro negócio, né? Eu acho que não foi medido, tipo, o tempo de coerência, qual a diferença do tempo de coerência, né? Que é um negócio que se pode medir, né?
Uhum.
Tipo, para ver quanto que é necessário esse pocket, né? E tinha uma outra coisa da sua pergunta, o que era? Da direção. Esse efeito que eu descrevi, né, de spin, no mais genérico, é um efeito que depende da direção do campo magnético, da frequência e da intensidade. Eu só descrevi a dependência na intensidade do campo magnético, né? Então, se você tem criptocromo na célula, né, assim, isotrópico, em solução, é possível ver um efeito, né? E é isso que eu acho que estão vendo os experimentos que as pessoas fazem em célula, em pratos de célula, né? Só que e esse é um tipo e o que eu vou falar agora não foi feito ainda. A ave usa mesmo o criptocromo para migrar, isso significa que o pássaro tem que conseguir ler direção. E ler a direção do campo magnético só é possível se o criptocromo tiver, né, fixado mais ou menos em alguma coisa, provavelmente uma membrana, né? E tem uns papers teóricos que falam que os criptocromos fixados numa membrana, e parece que o criptocromo é realmente fixo, pelo menos em pássaro, né, numa parte, no cone ou no rod das células do olho do pássaro, né? Que o criptocromo, se fixado daquele jeito, o movimento térmico dele não é forte o suficiente para destruir a informação de direção. E a dependência desse efeito na direção, até onde eu sei, nunca foi medida. E é uma das coisas que a gente quer fazer. A gente está montando um microscópio de TIRF, né, Total Internal Reflection, que a gente vai colocar os criptocromos todos para cima, né? Assim, absorvidos numa camadinha de ouro, né? Porque se sabe que se fixa bem. E a gente quer ver se a gente consegue modificar o campo magnético e ver uma mudança no estado do spin pela fluorescência, pela autofluorescência. Deu? Não deu? Outra.
Acho que eu devo entender um pouco. É bem legal. Muito obrigado pela resposta. E eu só queria perguntar só uma última coisa. Você sabe qual o par de moléculas que vocês estão usando dentro do criptocromo? Porque eu vi que ainda tem muita discussão se é a flavina e triptofano ou ácido ascórbico. Aí eu queria saber o quanto você sabe do que você está trabalhando.
Então, não se sabe, né? Nas nossas simulações, na nossa simulação na QuBiT e nas simulações standard, se acha que é a flavina com os triptofanos, né? Mas tem vários outros. Deixa eu até mostrar, se vocês tiverem um tempinho aqui, deixa eu mostrar um slide. Eu acho que a pessoa que está aqui, que é um cara super jovem chamado Daniel Ketnig, ele está na Inglaterra, em Exeter, né? Ele trabalhou muito tempo com o físico teórico chamado Peter Hoare, de Oxford, que é quem propôs, ele estudou bastante esse mecanismo, né? E olha esse diagrama do slide é meio poluído, mas tudo bem, deixa eu explicar. Aqui, às 9 horas desse reloginho, você tem o triptofano e a flavina, né? Esse ciclo é o ciclo de redox da flavina, né? O par de elétron que se acha no mecanismo standard, que é o responsável pelo efeito, é esse daqui, que está, sei lá, 11 o'clock aqui, 11 horas, né? Isso é o standard. O meu colega de Exeter, ele acha que, na verdade, não é esse daqui que as pessoas veem o efeito, né? Porque, aliás, tem uma disparidade que o efeito que se vê é mais forte do que o efeito que foi calculado, né? Então, o que o meu colega acha é que, na verdade, o que está acontecendo possa ser um efeito de campo magnético nessa combinação aqui, às 6 da tarde, nesse reloginho, que envolve não só o triptofano e a flavina, mas também os outros radicais, né? Então, tem vários tipos de modelo, né, que estão em jogo. E a gente acha que com os nossos aparelhos, né, que a gente vai conseguir fazer, assim, a gente vai conseguir sistematizar isso, né, porque a maior parte dos experimentos hoje em dia é feita em laboratório, assim, wet lab, né, de biologia, o que não tem problema nenhum, né? Só que, assim, só que a gente quer trabalhar num espaço que não existe, que é um negócio mais high-tech, porque a gente acha que precisa para ter alguma certeza mais se é quântico ou não, ou que parte da quântica que é.
Muito obrigado, Clarice.
Tá. Perguntas? Eu ia fazer uma pergunta, uma coisa assim, você viu o efeito de absorção de um fóton. Existe alguma importância quando absorve 2 fótons pela mesma molécula?
Não, várias coisas. Não sei no que se relaciona ao criptocromo, né? Uma coisa que eu lembrei que talvez seja interessante eu mencionar. Tem muita gente em biologia que está ficando super empolgada com essa história de microscopia de fóton gêmeo. Que assim, tem duas modalidades, mas assim, tem uma modalidade de microscopia que chama absorção de 2 fótons. E daí, que daí permite a você ver mais fundo no tecido, whatever. Mas o que algumas pessoas estão tentando fazer é microscopia a 2 fótons, mas com fótons emaranhados, com fótons gêmeos, né? E aparentemente tem algumas vantagens, não sei o que, da absorção, e isso é a única coisa que eu sei dizer sobre 2 fótons, que não é o que você perguntou, acho.
Tá. Não é essa coisa. Tem uma pergunta do professor Antônio Rion. Professor, pode fazer a pergunta? O que eu falo?
Não, eu faço, Orlando. Clarice, meus parabéns, né? Muito legal seu seminário, né? Show de bola, né? Desculpa a minha ignorância, né? Eu sei que o alinhamento de proteínas não é uma coisa simples de ser feita, né? E eu gostaria de saber como que vocês fariam, né? O substrato seria só uma monocamada do material alinhado, né? Principalmente o alinhamento. E o alinhamento, ele é importante para vocês?
Então, para esse experimento, sim. Se a gente quiser ver o efeito da direção do campo magnético, sim. A minha resposta para essa pergunta é assim, eu também não sei o que fazer, é por isso que no meu time tem gente que é físico, tem gente que é químico, tem gente que é engenheiro, né? Um dos meus alunos é químico, e assim, e ele sabe como fazer essas funcionalizações, não sei o quê, né? Não me pergunte, desculpa.
Não, tranquilo.
Se você tiver curioso e você quiser alguma coisa, manda um e-mail para mim que eu passo para ele.
Tá legal, eu escrevo sim, obrigado.
Eu acho que o Cauê tá faz tempo com a mão levantada.
Cauê, pode falar, Cauê. Desculpa, me desculpa.
Nada, professor. Professor, obrigado. Primeiro, agradecer pela palestra, foi muito interessante, tem mais informação sobre isso. Acho que a minha pergunta vai um pouco no contexto do professor Ryu, que é um pouco isso, como é montar esse time que é mais multidisciplinar, como vocês dividem as tarefas? O pessoal de física se dedica mais à física, a montar o experimento, a fazer os testes de setup, e aí você tem, integrar cada pessoa é multidisciplinar no time, ou é um time multidisciplinar e cada um tem suas especialidades? Assim, como você está lidando com isso?
Isso eu não sei, porque a gente está ainda começando e não é todo mundo que está no laboratório por causa do Covid. Então, agora no laboratório está mais a turma que está montando o microscópio, né? Mas assim, eu acho que é um desafio para todo mundo, né? Tipo, assim, você tem que ficar confortável com o desconforto. Assim, 50% do tempo eu não sei das coisas que o biólogo está falando com o químico, né? E assim, que a coisa legal é a coisa difícil e legal dessa tarefa, né? Que eu estou aprendendo o tempo todo, eu estou ensinando o tempo todo, mas assim, mas eu não sei como que você faz o alinhamento da proteína, né? E né? Porque em que lugar que mora uma pesquisa assim? Mora no departamento de física, de química, de engenharia elétrica, né? De biologia, de medicina? E é complicado, né? É complicado, não só isso, mas assim, como recrutar de todos esses departamentos, né? Como fazer para uma pessoa falar com a outra? E a gente está aprendendo, né? Assim, como falar na linguagem da outra pessoa, né? E outra coisa, para conseguir dinheiro de pesquisa é mais difícil do que o normal, porque primeiro que se você botar tipo Quantum Biology no Google, o hit número 13 é tipo Quantum Healing, né? Então as pessoas já acham que você é completamente fora da caixinha. E daí as agências de fomento também não sabem se é um treco meio louco, se é de verdade, né? Os físicos são os piores, desculpa, mas os físicos são os piores. Algumas vezes os físicos nem querem engajar isso. Quanto mecânica quântica a temperatura ambiente, você tá louca, né? E daí os biólogos, assim, eles falam, a gente já faz fluorescência química há trocentos anos, por que a gente quer um negócio que mostre o spin? Né? Então tem toda essa parte da nossa agenda, como um grupo, de disseminar, né? Disseminar o que é a biologia quântica e, né, para tentar normalizar onde a gente está e ter comunidade também, né? Porque hoje em dia, isso é uma outra coisa que eu vou falar, eu não fui perguntada, mas eu vou falar. Hoje em dia, a biologia quântica é um negócio de professor mais velho, né? Normalmente, os professores no fim de carreira, 1/3 do fim da carreira, eles já estão estabelecidos, já têm dinheiro, então eles se sentem confortáveis de pivotar para a biologia quântica e fazer esses experimentos mais novos, mais diferentes, né? Mas tem uma bruta barreira para a pessoa mais nova, né? Começar num negócio de biologia quântica, né? Então, eu acho que tem um negócio de treinamento das pessoas, da próxima geração, para normalizar esse campo e fazer ficar mais novo, né? Pronto, parei. É o final do...
Obrigado pela resposta, professor. Eu não sei se tem alguém mais para fazer pergunta. Eu acho que eu queria fazer só mais uma pergunta, professor, um pouco mais técnica. Não sei se a gente tem tempo.
Se eles deixarem, sim.
Acho que a questão é assim, é só curiosidade mesmo. Assim, com a história da medicina e da biologia assim, já mais antigas, eu imagino que você já deve ter tido experimentos do tipo, eu vou criar uma cultura de células ou uma cultura de animais expostos a um campo magnético forte ou muito fraco, como foi o caso feito ali. Mas quais são, tipo, eles são sistemáticos suficientes? Dá para olhar para esses experimentos mais antigos e falar, e a partir deles descobrir onde olhar por fenômenos que podem ter algum grau quântico ou dependência magnética dentro do organismo? Assim, tipo, quão possível é fazer essa coisa de olhar para o passado e conseguir ver novas linhas de pesquisa?
Então, é difícil porque as coisas não são muito sistematizadas, né? E daí tem, assim, tipo, o campo magnético a 5 Hz e, sei lá, 1/3 de Tesla faz mal. Daí o campo magnético a 200 kHz e não sei o que faz bem, né? Então é muito louco, né? Que tem dado em todas as direções, né? Tipo, tem estudo falando, o campo magnético ferra com as suas células, o campo magnético diminui o tumor do câncer, né? E todos os parâmetros são diferentes, não existe sistematização. Então, assim, o que eu acho é que, assim, existe coisa, né? Eu acho que dá para olhar para o passado e falar, nesse dado eu confio que eles viram alguma coisa, nesse não tanto, mas assim, alguns eu viro e falo, tem um meta-estudo, se você me mandar e-mail, eu passo para você, um meta-estudo desse negócio de campo magnético, e tem uns negócios que você fala, pô, acredito, né? Acredito que foi visto um efeito. Mas não é sistemático, e essa é uma das coisas que a gente acha que a gente pode conseguir chegando, olhando para a coisa de um jeito, né? Fazendo um experimento que, vamos fazer um raster aí do campo magnético de meio em meio ângulo. Vamos fazer um negócio, sabe? Eu acho que a gente vai ter um pouquinho mais de liberdade para sistematizar isso, que é o importante, que está faltando, o que você tem razão, absolutamente razão.
Certo. Perguntas? Perguntas? Acho que não. Então, queria falar, queria agradecer muito a disponibilidade da Ana Paes aqui. O professor Doreto, Mergado Doreto, né, ele é organizador desses colóquios da pós-graduação, né, e tem nos ajudado muito para manter os colóquios, mesmo no meio da pandemia, que é uma coisa complicada, né. E daí queria agradecer também a sua disponibilidade em dar esse tempo para falar com a gente, né, que é interessante para os alunos, né? Aqui tem muitos alunos estão no início da pós-graduação, então a gente vai ter uma coisa interessante, até para achar tópicos para onde estudar, uma coisa e tudo, né? É uma coisa bastante interessante. Então, eu queria agradecer isso, né? E está aberto as portas, se tiver talvez uma outra hora, tipo assim, quando acabar a pandemia, vir para o Brasil, passar de Jundiaí, passar para Campinas, essa coisa.
E vocês também, por favor, mantenham contato. A gente tem um monte de... A gente tem contato com um monte de gente no mundo inteiro. Se vocês quiserem, se alguém tiver alguma ideia, quiser discutir, propor coisa, vier trabalhar com a gente, manda um e-mail, a gente começa a conversar, né? E obrigada de vocês terem dado esse espaço para alguém falando de biologia quântica. Para mim é muito, eu fico muito grata que o departamento de física abriu as portas, porque não é um negócio muito comum. Então, muito agradecida pela confiança, né? Pela confiança.
Certo. Eu queria pedir, então, todo mundo dar uma salva de palmas para a professora. Certo? Então, muito obrigado.